Welcome back. This is the part where I will introduce to you several types of nucleotide databases. Let's begin. In the previous topic, I have introduced to you about the structure of databases. In this topic, we will be looking directly on the types of nucleotide databases and learn more about GeneBank file format. When we talk about nucleotide databases, what comes to mind? Well, I'm sure you have understood the meaning and purpose of a database. So a nucleotide database serves just that. It is a collection of DNA sequences from several sources, for example, genome data, gene, and transcripts. These type of DNA sequences provide the foundation for the scientific research and novel discoveries. I will briefly introduce to you some examples of nucleotide databases. GeneBank is a genetic sequence database that contains a collection of all available DNA sequences submitted by researchers. It is part of a consortium known as NCBI. NCBI collaborates with DNA Databank from Japan and the European Molecular Biology Laboratory together with the European Bioinformatics Institute. Their main collaborative work is to provide genetic information that covers the wide spectrum of raw data, for example, raw sequencing data, and many more. This collaboration is known as the International Nucleotide Sequence Database Collaboration. Meanwhile, the nucleic acid database is another type of nucleotide database. It is a portal for three-dimensional structural information about DNA. You can use this database to study the structure and functional characteristics of a nucleic acid. The European Molecular Biology Laboratory, together with the European Bioinformatics Institute, maintain a collaborative work at Hingston, UK. EMBL itself has another 4 different research laboratories in Germany, France, and Italy. Specifically, the European Bioinformatics Institute provides a platform for big data query in biology. There are many types of DNA sequences, and the most common ones are exons and introns. All these sequences can be retrieved in the GeneBank. NCBI was created in 1988 as part of the project at National Institute of Health. It is a center that created public databases for scientists. Their main research is in bioinformatics. The main function of this institute is to disseminate the biomedical information to the public. Under the NCBI, there are 2 types of databases, primary and derivative databases. A primary database contains the original submission from researchers. GeneBank is an example of a primary database. Derivative databases is a type of database that is derived from the primary database. This type of database contains unique information about a specific domain, or species, or disease, or etc. A good example of a derivative database is a reference sequence database. Let's take a closer look at how these 2 examples of databases could be different. Much of the difference between GeneBank and reference sequence is revolved in the type of information they specifically carry. For example, ref sequence database contains curated data or information that is always updated. This is because this database is controlled by the NCBI itself. RefSeq contains only unique sequences which are of researchers' interest. For example, a reference gene sequence in human genome. One of the characteristics of a good database is that each database record carries a unique identifier. Since RefSeq is a non-redundant collection of sequences representing genomic data, transcripts, and proteins information, all records were properly labeled or annotated. All these records are grouped under different types of groups, and each group of sequences has a different category of accession numbers and description. For example, sequence with categorical NC is a collection of complete genomic molecules. Regardless of the type of sequences both GenBank and Raster contain, all sequences are stored in a common file format, as shown in the next slide. Let's take a closer look. This is the file format of sequence at the NCBI. Basically, there are four main sections: title, header, feature table, and sequence. Under the title section, you will read about sequence description, accession number, and links to either FASTA or graphic information about the sequence. Under the header section contains detailed information about the record. For example, the accession number and its current updated version, organism, genomic location, and so on. When you want to find a specific detail about exon or coding region, for example, feature table provides the answer that you are looking for. Under the last section, which is the sequence, you will see the complete stretch of either a DNA sequence or amino acid sequence. Records are grouped for various reasons. The main reason is to allow users to be fully taking advantage of this database using specific sequence identifiers. In order to further understand on how to read these sequences, a researcher and people like you need to be able to know the meaning and purpose of these identifiers as shown in the slides. For example, by understanding the meaning of protein ID, we would be able to know what a particular record contains, some structure, and functional details that are important to us. In this chapter, I have uploaded a video tutorial on how to see and understand this section more. In this topic, you have learned about several types of nucleotide databases, the primary and derivative databases and their differences, and GenBank file format and sequence identifiers. Thank you very much.